Hello, everyone. My name is Kainelainen of University. My supervisors are Igor Drobyshev of Swedish University of Agricultural Science and as well, and Fabio Gennaretti. And today I'll be presenting preliminary results of my part of my PhD. The theme is wood volume accumulation rates of balsamifera after two different designs of low intensity cutting in boreal mixed woods. As we've introduced already to problems of modern forest management, we know that there is a need to diversify the cutting practices, particularly partial cutting. And besides, there is also a need to manage mixed wood forests composed of different species. So today I'll be presenting my results. My objective of the study is to study the effect of partial cuttings in boreal mixed woods on the growth rates of balsamifera, and also to assess the effect of interspecific and interspecific competition with trembling aspen as hardwood species, which is commonly growing along with balsamifera in boreal mixed woods, and to assess its effect on the growth rates of fir. Here is the study area. My study has been conducted in Abitibi region of Canada. It's field forest. And here is the map. So I have several experimental units for control without any cutting, and four experimental units with partial cutting. There are two different partial cuttings. The first one is so-called dispersed cutting, and the second one is block cutting. Dispersed both cutting practices, where percent of removal of basal area was around 40%, so it's low intensity cutting, and they differed in design. So dispersed was the removal of around 40% trees. So it was not aggregated along the skidding trail of machinery, while gap cutting or block cutting, the cutting was aggregated in 400 square meters gap along the skidding trail of a machinery. The data collection has been performed in summer of 2020 within these experimental units. So experimental units are circular plots of 400 square meters area. I have conducted detailed inventories with calculation of density and basal area within these experimental units. So basically I measured the DBH of these trees and quantity of them as well. I have sampled around from 30 to 50 balsamifera and trembling aspen cores from each of the experimental units, and besides, from 5 to 7 residual balsamifera trees have been selected for competition assessment within these experimental units or in nearest proximity. And around these selected trees, within the circular patch of a radius of 7 meters, I have assessed the competition. But I will explain further how it was done. And these residual trees were sampled for stem analysis, where you cut down, and wood cookies, as here on picture, were extracted starting from the base level, ground level, then at the breast height, and the next every 2 meters until the stem reached 5 centimeters diameter at the crown. So this is how it was done, since the sampling took place on so-called safe-free site. We had to cross a river, and the next picture is basically how a sample tree looks like. Here I present the results of inventory of which experimental units. As you can see, two species of interest are highlighted here. Balsamifera is pink and green is trembling aspen. You can see that they are basically strongly dominated by these two species. All control plots and cuttings. Some have a higher share of balsamifera, while ours have more trembling aspen. There are also our species like paper birch and spruce, for example, but the share is quite small. The chronological analysis was done in standard software as QRecorder, Seed and Draw. Cross-dating was done in Cofetia. And the calculation of wood volume from wood cookies was performed in R package Triglia. And here are the results. So when I calculated the wood volume, I encountered a problem that the data was not really comparable, since trees originating from control plots, they were generally larger and older than trees from partial cut practices. Here you can see the graph of volume increment and the years on x-axis. And by 2000, so the cutting happened in the winter of 2000, you can see that the wood volume accumulation, wood volume increments from trees originating from control plots, they were higher than from trees with two different cutting designs. And here to the right, there is a box plot of age of trees from three different cuttings and also the DBH. So even for the age from dispersed cutting and control plots was comparable, trees from block cutting were significantly younger, and also the DBH from both control plots was lower than the DBH from trees from control. So here I present the mean volume increment for different time periods. So I divided the time by 5 years time periods, starting from 5 years before the cutting treatment to see how trees were growing before the cutting, and then for our time periods up to 2020. As we can see here, the wood volume increments will gradually increase from trees from all treatments. However, since wood volume was higher from trees from control plots was already higher, as they were like bigger and older, they accumulate more wood volume. And there is also the last time period, there is also slight decrease for trees from all treatments. What is more interesting is to look at the relative wood volume increment increase. As you can see, the relative increase to the previous time period is higher for cutting practices than for control trees. And this trend is retains for basically for first 10 years. It's especially noticeable. Then there is also it's increasing, but slowly. And the last 5 years there is a decrease. But higher increase in volume increment may also be explained by younger age of these trees. So maybe young trees may accumulate more wood volume as well. So the main part of my study here was to look at the competition in this In order to do that, I calculated the NCI, so-called NCI, competition index, which were detailed explanation of competition indexes is provided in the paper of Rowan and Kuulainen, 1997. Basically, how it was done, it's here to the right, you see a picture. There is a focal tree highlighted in red. It's a tree which was sampled for stem analysis and cut down within a circle of radius of 7 meters. All trees of merchantable size were assessed. The DBH was measured for all these trees, and also so was measured the distance between the focal tree and between all the neighboring trees. And I have tested 4 different types of NCA competition indices for main types, and they all actually computed by there is a sum of either diameter of of neighboring trees divided by the distance, or the relationship of diameter of focal tree to, or neighboring tree to the focal tree divided by the distance, or arctangent. This is to assess the horizontal angle between the trees. And I have also applied the coefficients to each of the NCA as 0. It means that there is no effect of the diameter or distance. 1, if there is a straight relationship, and 2, if there is a for square, yes, for squared effect of diameter, for example, or square root effect of the distance is coefficient of B of 0.5. And I assessed all these competition indices, and the predictive they were measured by assessing the predictive power by calculating a Kaika information criterion, ASC, and in the additive model. And here is a general equation of a model. So it was response variable was square root of mean volume increments for each of these 5 time periods, starting from 2000 to 2005, 2006 to 2010, and so on. There was an interaction between competition index and treatment effect, and also we were split by time, by 5 time periods, and I added a smoothing spline as an H, since it wasn't equal between the plots, and reduced the number of dimensions, K index, by 3 to make as the default level was the curve of smoothing spline was too wiggly, so it reduced it to 3. And I found out that the best equation with lowest AIC score was the one with relationship with a sum of relationship of diameter of neighbor trees divided by diameters of focal trees, and this is all divided by distance. However, the alpha, it was a squared relationship of diameters, but the distance was not important, so the coefficient was 0. And after that, yeah.
Sorry, Ari. You will have to conclude.
Yes, okay. So here I plotted the estimates of this model, and important estimates, not all of them. And these are basically the results. So we can see here that interspecific NCI has a stronger negative impact if it's the NCI of time and NCI. So it was expected as interspecific competition is has more negative impact on the growth rates of trees than the competition index of aspen for fir, and also cutting treatment okay, so the cutting treatment has a negative effect for the first 10 years following the treatment, and when the competition indexes they become mostly negative. For balsam fir, interesting competition, there is no, the picture is not really clear, but for competition index of opposite species, we can see that actually trees from dispersed cutting treatment, they have a positive estimate. So it means that the effect is, a stronger negative effect is found in control plots. So cutting reduces the effect of interspecific competition. And that's all. Thank you for your attention.
Can you hear me?
Yes.
Oh, hi. When I was looking at the tree cuts, I was thinking like, wow, they're really nice and they would be so great for isotope analysis. You know, where you can have laser ablation where there's no more sample processing needed. And then at the lab, they'll run a laser over it and then they'll have really high detailed information on carbon isotopes, which might give like even more supportive evidence to what you found as well with the accumulation. Have you considered isotope analysis?
Isotope analysis, I haven't done it, but possibly, I mean, it possibly can be done, but of course it's more complicated. It requires like equipment, and it's more hard to implement.
Okay, thank you.